morning and welcome to today's GCSA webinar, Mastering Annual Bluegrass Weevil Management, Insights from the Experts, presented by Syngenta and taught by Lisa Beirn, Ph.D. and Ben McGraw, Ph.D. My name is Lisa Wick. I'm Senior Manager of E-Learning Programs here at GCSA AHQ in Lawrence, Kansas. I'll serve as today's moderator. Before I turn things over to our presenters, I want to thank Syngenta for presenting this event and helping make our live webinar education available as a free member benefit. Syngenta proudly serves as a GCSA Platinum partner, building on decades of industry collaboration and innovation. They believe in empowering turf professionals with more than just trusted products by providing comprehensive support through technical guidance, advanced educational resources, and digital solutions on greencastonline.com. Their integrated approach helps professionals achieve results while driving the industry forward. We thank Syngenta for their support of the live webinar programs as well as other GCSA programs. A couple of quick housekeeping notes. We are recording today's event. You'll receive access to that recording via follow-up email. We do have a handout available in the GoToWebinar control panel. If your control panel has minimized, you'll see a little narrow strip with an orange rectangle at the top. Click on that, and that will maximize that control panel. You'll be able to access the handouts. You'll also see there the question and answer box. You can type in questions as we go along today, and we will take those in due course. If you want to ask your question directly on that same tab, you'll see a little raise hand icon. You can click on that, and that will let us know that you want to be unmuted when we pause for questions. Lisa Beirn is a Technical Services Manager for Syngenta in the Northeast and Mid-Atlantic U.S. She earned her bachelor's degree in Biological Sciences from Cook County, Rutgers University, and her master's and Ph.D. in Plant Biology from Rutgers University. While at Rutgers, Beirn conducted both applied and basic research on several important fungal diseases of cool season turf. Her work has led to an increased understanding of pathogen biology, including the taxonomic reclassification of dollar spot. Ben McGraw, Ph.D., is a professor in Plant Sciences Department at Pennsylvania State University. He obtained a Master's of Science in Entomology from the University of Massachusetts Amherst and a Ph.D. in Entomology from Rutgers University. McGraw's research focuses on understanding insect ecology and behavior to improve management programs. For the last decade, he has applied this focus to improving the control of annual bluegrass weevil, about which we will hear more today. I want to thank you for joining us. I want to thank Lisa and Ben, and I'll turn it over to you.
That's got to be an old bio, Lisa. I think I've been doing this more than a decade, and lots of love for Rutgers, and I'm going to say the talent portion of this competition here today is Lisa Beirn, who was a rock star at Rutgers. So what we'd like to do in this seminar today is kind of lay the groundwork for this pest, because I know that there are people on this call who have varied experience with this insect. So probably a lot of first timers or new to the pest. And I think this is one that's a little bit different from most of the insect pests that we see. So making sure that everybody's on the same page and understanding the basic biology of this insect is my job. And as I mentioned, Lisa is going to bring it home with kind of the new innovations out of Syngenta. So I know I'll be taking a lot of notes in the second half of this. But the annual bluegrass weevil, by far, is the #1 pest that I see in the Northeast, and really because if you think about it, it's the ultimate pest. It really does some things that are different from our other insect pests. It keeps us on our toes. It has the potential to be far more destructive than just about any turfgrass insect pest that I see in my travels. But its biology is one that really makes it such a difficult pest. We have to contend with this multiple times throughout the year as it goes through different generations. It has what we refer to as cryptic stages, or stages that are hidden within the plant. And if you think about some of the areas it can attack, like putting green surfaces, albeit rare, those are very small plants. So very small insects with hidden stages inside the plant makes it a real challenge to control. And we've been doing battle with this insect for decades upon decades. As a consequence, we can see that some of our products have stopped working well, or that the insect is the ability to evolve resistance to things like our insecticide application. So for those of you joining us who are trying to get more information about this because you're dealing about it with the first time or first couple of seasons that you're dealing with it, and you want to know more about it, it is a full court press pretty much all season long. There are certain times where we're going to be really focused on trying to detect this insect in the landscape. And there are times when the populations are going to be depressed, but it's no time to let up, even in those summer months where we might not see much activity. So, just a little bit of background about this insect pest. As I mentioned, it's been around for several decades, mostly in the northeastern United States. So for much of the beginning, from its initial detection in the late 1950s on Long Island, New York, it was pretty much isolated in the tri-state area, New York, Connecticut, and New Jersey, and a little bit after that, it started to expand into eastern Pennsylvania. But for a very long period of time, it was isolated in a small area. This insect is not a big flyer, so it primarily moves around by walking, and possibly we move it around through some of our, moving the sod and whatnot. So it was very contained in a small geographic area for a very long period of time. This paper that I have referenced in the left-hand corner of the screen was some early work that was done by researchers at Cornell to really describe some of the stages of the insect, as well as some of the other weevils that we see on golf courses. It's something I refer to quite frequently. Those same researchers were, Cameron and Johnson were, instrumental in trying to come up with ways to control this pest. And what we see in the early days, in the very, very early days of the detection of this pest that was causing this die out on the edges of turf areas like fairways or possibly collars, they just tried a battery of different insecticides against it. So right from the beginning, we have been very chemically focused on this pest. In that early study, that you can see in the left-hand corner, they were trying very harsh things. It's interesting to know that things like DDT did not work very well against the weevil. You can see just how difficult it is to control something like a beetle. Throughout the years, we started to kind of see that even those approaches were kind of had some chinks in the armor and started to break down. And really the work that was done by my former master's advisor, Pat Bidham, as well as her advisor, Toshiro at Cornell, really set the early work for studying the nuances of the biology and the ecology of the insect to help us better understand how these chemicals could be integrated better so that we could get better control. And so I would say that was really kind of the only early work. We're out of a couple of labs in the Northeast, and things seemed pretty good for a little while. And then what we can see is that management, not of the insect, but really management of surfaces really changed in this period. From the late 70s, you think of the release of the stimp meter and the impact that it had on fertility, mowing equipment, the thinner bed knives on real mowers allowed us to mow much, much shorter. So this is a slide from my turf grass management professor at Rutgers, Rich Early, shared with us is, the difference between a fairway in the mid 70s to the 1980s really dropped dramatically. And in this time, what really became noticeable is a bunch of different insect pests that were previously probably less apparent in their damage. So annual bluegrass weevil absolutely flares up in the 80s into the 90s and 2000s. So a lot more stress on the plant. However, at the same time, we see really the start of what I think of as modern insecticides coming to light. In the 1990s, the pyrethroids enter the market, at least the photostable pyrethroids, derived from pyrethrins. And what we can see is that these were very, very efficacious. They were excellent at controlling insects that might be active on the surface or insects that they could come in contact with. Widely popular class of insecticides that we see, really revolutionizing a lot of agricultural commodities. What ended up happening in the 2000s is some unique things with the change in the insect and its ecology to some extent. In the 2000s, what we started to see was damage to creeping bentgrass. Primarily, it had been an annual bluegrass pest. There was very little mentions, possibly different species of weevils on golf courses that were attacking creeping bentgrass. But as this insect continued to expand its distribution, moving into more southern areas, we see a lot more reports of damage to creeping bentgrass. At the same time, what we're starting to see is a decrease in the efficacy of the pyrethroids. And what we suspected is that these two things are happening independently, that all of a sudden, people who have been misusing pyrethroids, too many applications per year, are starting to see breakthrough in those products. Really, in the mid 2000s, we see a great deal of innovation from our industry partners that really kind of continues today, which will be the second half of this talk. We see a real proliferation of different classes, a lot more selectivity in these products, a lot better environmental profiles. And what we see is rapid development of these throughout this period. So it's not all doom and gloom through this era, but that's really kind of the basics of the history of this insect today. But probably the most current history about this insect is where is it going to show up next? Again, what you can see is that for many years, this insect was locally distributed, kind of isolated within the Northeast, surrounding the New York metropolitan area. And if you're watching this webinar today, you might be in these states with the dark blue. We've seen a continual radial expansion of this pest outward, both to the south and to the west, but also to our Canadian neighbors to the Northeast as well. So in 2016, this was really the first time that we saw a great jump of the insect outside of its distribution, and we have some pretty good evidence, working with agronomists at the USGA, that this was an incident where the insect was moved through sod from New Jersey to Arkansas. So that made a ton of sense. But over the years, what we saw, especially in 2018 and 2020, is a great proliferation of reports coming out of Kentucky, Wisconsin, Indiana that did not seem to be always associated with the movement of sod. Whether this insect had always been there and it had just been apparent, or if another stressor showed up and then people decided to look for these insects, we just really don't know. It's quite a mystery how these things that don't move great distances are all of a sudden showing up in these far reaches outside of the Northeast. Most recently, we can see the western edge of the distribution showing up. I think this is really an interesting area where some labs, my colleagues at NC State, are really working on characterizing what these insects are, if they are in fact annual bluegrass weevil. I think we can definitively say that some of these populations are, but we might also be seeing some other weevil species confusing the matter to some degree. So stay tuned on that. But what we see is most of these new reports are really on the western edge. The ones in the southeast, like Georgia and South Carolina, I think those are very close to the western North Carolina border, where it's a little bit cooler and the climate is more similar to what we would see in the Northeast. So a lot of detections all of a sudden in faraway places. Hopefully this is just an example of good scientific communication and people are more aware of the problem. All right, so we conducted a survey in 2017 to really see what impact ABW has on management practices, on budgets, on control theory. And again, this is 2017, very different from today. But we surveyed it and got quite a good response from superintendents throughout the distribution at that time. And what superintendents were saying is that they would spend almost 4 applications solely on annual bluegrass weevil. What we will see is that this is one of our first insect pests that come out in springtime when no other pests are around, where we can't match up our control with these other pests. And so, it does demand quite a bit of effort to control these populations below damaging thresholds. Now, this is an average. There's quite a bit of variation, both on the high end and the low end. We also asked about, how many areas on the golf course do you experience damage? And these are, pretty much roughly about 13 acres of damage that we would estimate from that could be under attack from the annual bluegrass weevil. So the average control at that time, so we asked for budgets, not only maintenance budgets, but also insecticide budgets, but also ABW application expense. And it was, about $9,000 to control this insect. Now, this is a large problem that I see with people who have annual bluegrass weevil for the first time, and we start talking about how we control this multi-generational pest. This is a big sticker shock for many. So $9,270. I will also mention or reiterate that this is 8-year-old survey. And this is before some of our more expensive insecticides that hit the market. These products, I believe, has superior efficacy, but there are several insecticides that would actually cause this estimate to be much, much higher. So I suspect it would be much, much higher for most of your operations out there. All right. So today's outline, what I thought we'd do, just so that we bring along everybody on the ride, is to really kind of go over the simple aspects of this insect in a Cliff Notes section. I'm dating myself there, that younger people on the call don't know what Cliff Notes are. It was the analog version of ChatGPT, to put it briefly. From this, once we understand the biology, and it is critical to understand the biology about what's susceptible, what's not, what can be controlled, what can't be controlled, then we can talk about control theory, about the approaches that we take or the approaches that I would recommend, whether you had a light infestation or a population detected for the first time, or you have something that's rather established. We also have to talk about monitoring if we're talking about control theory, about how we time these things for the different stages that are susceptible. And then I get to turn it over to Lisa, where I'm also very interested to hear about what they've been developing at Syngenta as far as industry innovation. So to jump right into it, this has got to be one of the most attractive insect pests that is out there in the world. This is a very sleek looking beetle, very, very tiny, roughly about 4.5 millimeters in length. That is incredibly, incredibly tiny. It is a weevil. So what it has is this beautiful snout at one end. The female will chew a notch into the stem of the plant, and she will insert her eggs inside the plant. So very, very tiny, tiny insects that will emerge and feed within the plant, at least initially. What we'll see is, if I can just back up and use my laser pointer. Oh, Lisa. Probably too complex. The first 3 instars that we can see, hopefully you can see my cursor moving here, is these 3 instars are located pretty much inside the plant. We refer to this as cryptic. They're hidden stages inside the plant. They're well protected from the environment. They're also well protected from most of our insecticides. Oh, look, there he goes. All right, thank you, Lisa. These 3 inside the plant. Once they're too large to fit inside the plant, they're going to feed externally. Unfortunately, they feed externally on the crown of the plant, causing the death of the plant. This final stage is the pupa, which is a dormant, non-feeding stage that is going to transition into a new adult. This orangish appearing adult is just newly formed. Its exoskeleton has not hardened up, it hasn't melanized, it hasn't darkened, and you can often see these on the surface as well before it fully gets its scales, like this older adult here. So those are the important stages, and you can see that they transition from inside the plant to outside the plant, from kind of innocuous to more damaging stages as they age. So the big thing about this insect, I think, one of the things that we kind of overlook is really its reproductive potential. So early reports in that early paper, but from Cameron and Johnson, would suggest that they lay between 12 and 20 eggs. And when we conducted a study several years ago where we actually tracked females over time, the average egg laying was somewhere between 60 and 90 eggs per female. That's quite a bit. What we can see is that they can lay for a very long period of time. However, most of those eggs are laid if we protect the female, and I'll show these data later. They have the ability to lay for a long period of time, but most of their eggs can be laid within an 8-week period. Still, very long period of time. What this all means is if you have a lot of offspring, some of those offspring are going to have just, superior genetics or the lucky gene that allows them to develop resistance to something like an insecticide that they've never seen before. So greater probability to evolve to whatever we throw at it. Now, the other thing, as I mentioned, it's really important in its biology, is that we have a bunch of cryptic stages. Cryptic meaning hidden within the plant. So not only the eggs, you can see the eggs up here, they're kind of protected underneath the leaf sheaths, probably provides a very nice moist environment for them to develop. And then I've highlighted here is the larva. This is an early instar larva where we're basically seeing it through that translucent leaf sheath. These are protected from most of our chemical insecticides as well. So they not only are small, because these are small plants essentially, they're also really well protected. So difficult to monitor, difficult to control at these stages. Now the late instars, too big to fit inside the plant, so they're going to exit the plant. They're going to probably feed quite locally. These are not very mobile larvae. They're going to feed, probably very close to where their mother deposited their egg. Unfortunately, they feed on probably the most important, I'm not going to say probably, the most important part of the plant, which is the crown. There's no regrowth from the damage of this crown. So if we have lots of population, a large population of fourth and fifth instars, we could expect to see damage being quite severe. All right, this is a pupa stage, a really beautiful stage. This is going to be in the couple 1 to 2 centimeters down into the soil, not very deep. It's going to transform from that worm-like larva into something like a beetle. At this time, you can see it's starting to develop its wing pads here. We can see its snout with its mandibles there, the chewing mouth parts. It's going to lay, in there, depending on the temperature. Most estimates in the textbooks would say between 7 and 10 days between that final instar and becoming an adult. This is not a target for our controls. What we see is that pupae and most insects are very well protected by this outer casing. It makes it very difficult to deliver insecticides into the body of the insect. All right, so I'm putting this up against my better judgment. I hate to use dates, exact dates. I'm going to give you a rough range because we probably have people from a great geographic distribution here. Again, I have growing degree days here on the bottom. We will talk a little bit about growing degree days. In the entomology world, or at least the ABW world, we use a March 1 biofix. So that's when we start recording growing degree days. We use a 50 degree Fahrenheit base temperature for development. Other than that, it's calculated just the same as any other growing degree that you might use in your operation. So again, against my better judgment, I have to have something for a reference here. I don't want you to get fixated on these growing degree numbers or the time frame. I'm going to give you rough estimates. The important thing is to look at how this insect develops throughout the year. The other important thing is to look at the relative sizes of these peaks and when they occur. All right, so the first thing that we see right out of the gate is that the adults will come out of overwintering sites. They will build up to a peak. So in this chart, this is a hypothetical population development if we did nothing. We did not control it. We just sent a nerd out like me to count bugs all day long. This is the kind of population development curves that you would get. You would see a massive increase in adults that as they leak out of overwintering, they build up to a peak and then they'd start to decline just naturally. You would see a huge spike in larvae and then that would decline naturally. So these are the overwintering adults that come out in springtime. They lead to these large peak in larvae. And if you look across the entire year, that's probably going to be the largest peak of larvae that you are going to see all year. This is the largest densities of populations that we see in springtime, and it's usually the most damaging. So this is roughly between, I would even say March in our more southern locales, all the way into June, probably mid-June in our more northern populations. What happens in summertime, so after we have this peak of first generation larvae, if we did nothing, again, we'd see damage around in this period of time. We'd see a huge peak of adults again. So these are the first generation adults from the first generation larvae. And then what we can see is that in summertime, our populations are depressed a little bit. This is unusual for insects that have multiple generations per year. You would think uncontrolled, the peaks would just get bigger and bigger and bigger. That's not the case. And what we see in the subsequent summer generations, we see smaller peaks of adults, smaller larvae. We have some theories about these things. A lot of it is anecdotal. But the important thing is these populations get less and less. In most places, we believe that we see 3 generations per year. Some northern locations has been documented 2 generations per year. That was done many, many years ago, probably needs revisiting. In southern populations, we just don't know how many generations they undergo. But the big point is big densities, big potential for damage earlier in the season. All right, so this is our main focus, is this first box right out of the gate. If we can do our best to flatline this population here, as you can see, it's like a conveyor belt. You're going to be making your summer populations much, much lower to begin, essentially, by essence. The other thing that you can see is this is a jumbled mess, and I will explain this jumbled mess. If we start, and this is the case where a lot of people are dealing with the insect for the first time, they call me in this period here. And they say, I've got damage, I didn't know what this was, did some internet reading, got your number, what can I do? And I say, this is really, we'll try to right the ship, but it's going to take a lot of effort to get this. It's really important to start off on a good foot in spring. Now, why is that? Do we have a question?
Yeah, I have a quick question. Sorry. I was trying to.
I heard your heavy breathing there, Lisa. Go for it.
It's so exciting. The overwintering, does that increase or decrease at all based on soil type?
Overwintering survival?
Yes.
Yeah, I don't know if anybody's done any overwintering survival based on soil moisture, soil type, or anything like that. I would say that most insects that overwinter, or native insects like this that live in temperate areas, are pretty well adapted to freeze thaw, lots of conditions, that lots of variability in winters from one year to the next. That is a good question. I don't think I really have the answer for it. We typically get that question a lot. What do you think this winter is going to do? This was really cold. And I would say that they're well adapted to. They don't go very far down in the soil, so I would suspect that it would not have a great impact on survival. Great question, though. Awesome. All right, so most of our insecticides, with the exception of one that I can think of, work on the nervous system of insects. So they're neurotoxic chemicals. And what we can see if we look at that group, if we group them all as neurotoxic insecticides, what can we actually control? The eggs are not going to be affected. We typically don't have products that control eggs or are ovicidal in their nature. We do have some products that we apply when the insect could be in the stem, but for the most part, and maybe we'll see later on in the presentation, most of these insecticides are not very effective at the early instars. It's interesting to me, this isn't a period where we see a lot of turf loss. The insect is boring up and down within the stem. You would think that would be death to the plant. But we don't really see any signs of damage at this period. So they're very well protected, and we're not really going after them with our targets, but we could apply something that has residual activity at this time period, meaning you apply that insecticide and you're just hoping it hangs out long enough for when they pop out of the plant in the 4th and 5th instars. So these crown feeders here, they're going to be the biggest larvae that we have. They're going to be feeding voraciously, especially the 4th instars to get to that last instar, the 5th instar. So these are the damaging stages. We need to be really precise with most of our insecticides. And once it goes into this pupa stage, that window closes again until they become adults and we can control adults. So really the 2 stars here are the 2 stages that we really can target effectively. So it's not about timing here, it's what stages are really effectively targeted. And it's really the ones that are naked, the ones that are out there that can be exposed to the insecticides, that the insecticide can come in contact with the insect and enter the body somehow. All right, so what the jumbled nest means is if we look at this snapshot in the summertime, maybe this is, mid-June to mid-July or mid-July to mid-August even. What you can see in this hypothetical population is this time of the year, we have every stage present. If you didn't take control, maybe you notice this population for the first time, you didn't take any sort of control whatsoever. You go out there with a sample, you take a cup cutter sample from an area, and you could probably pull out every stage. So on the left-hand side, this photo here is just something, we took a zoysiograph plugger, a very small plugger, took it out of an area that had not been treated all year, and what we have is 4 instars right here. And there's probably some adults wandering around as well. So if I have 3 instars that are protected within the plant, and I applied an insecticide that could potentially control these 4th instars, that would be the only stage that gets touched. The next week, these 3rd instars would be 4th instars. 2 weeks after that, these seconds would be 4th. It would just be this endless conveyor belt of larvae entering the population. So we really need to have controls in place prior to this to at least compress or synchronize the larval population as it comes along. This is the case that we're going to have in the summertime. We're just going to have a jumbled mess, and it's going to be really difficult. It requires a bunch of applications to get ahead of it. So that's something that we want to avoid. Now, the first 1 to 2 applications of the springtime, and I'll go over control theory in a moment. These are going to be the most critical, I think, for the whole year. And I think in some instances, in some populations, might be all that you need for the year. Hopefully it's done in concert with scouting for the rest of the year so that you know whether or not you need to take that control. All right, so when we talk about programming for this insect, typically if we look from overwintering adults here on the left-hand side all the way to these crown feeding larvae, there's about really 3 big timings in the springtime. We'd have an adulticide that we'd apply against the adults, and that has many advantages. I'll talk about that in just a moment. We have some applications against early larvae, and why I have that asterisk there is most of our products do not last very long. And so if we're applying it at this timing and there's a whole bunch of younger stages in the plant, that can be difficult. Usually we're applying the early larvicide application to be present for when they pop out as plants as a crown feeders. We have several products that we can use once that insect is out of the plant, feeding in the soil externally. Those products need to work really fast. They need to be really efficacious as well to prevent any sort of turf loss. So you can already see the challenges. If this was white grubs, we'd be talking about probably 1 application per year. Chinch bug, we'd talk about applying a contact with it. This one involves a whole lot more really kind of deliberate actions, really kind of conscious, really well thought out programming. All right, so the other thing that I will say, and I'll show you some data slides in a moment, just know that control is not perfect with these insects. If adulticides were 100% perfect, we wouldn't have a problem. If any of these larvicides were 100% perfect, we wouldn't have a problem. So, we do have a little bit of lowered expectations when we talk about these things. It doesn't mean that controls can't be effective. What we're looking for is reducing populations below a damaging threshold, not total elimination. All right, so let's start in the beginning with the adulticide application. This is an application that to some is controversial, to others, I think, like myself, I think it's still an important aspect to minimizing our populations. These things are like mini tanks, and they start marching out, storming in their tanks very early in the season if we have these warm spring days. So what we can see is there are some challenges right out of the gate. 1, they're mobile. 2, they're very, very small, so it's often hard to detect. And then the third is the challenges with the environment. So you might have a population under this tree or in the tall grass, maybe in the bunker bank. And what you're going to see is insects are cold-blooded. So once it warms up to a threshold where they can move, they're going to start marching out. And as long as the temperature is conducive for movement, they can move. However, if it drops down, like we often see, large fluctuations in springtime temperatures, these insects might just go into suspended animation and not move until weather warms up again. So what we can see is, you know, people often jump the gun. They see those first arrivers on the fairways, and at this point, they're saying, now's the time to spray. Then it gets cold. That product wears off within 24 hours to 48 hours, and now all of a sudden you have them marching out again. So there is some nuances to when we time these things. It's not difficult, but I do see a lot of early spraying. That was a big thing for 2025 that we saw. Now, if we look at the whole buffet of insecticides that we have available to us as turfgrass managers, it's not a whole lot. We don't have that many classes. There's about 11 on this slide. And this is every insecticide. This is not what works against ABW. This is your insecticide menu for the United States in 2025. The main ones that we're going to see need to kill a beetle. That's a very difficult thing in the world of entomology. It needs to get through that waxy exoskeleton and somehow get into their body. That's a tall, tall order. The other thing that we see is that these beetles, at least in overwintering, as they come out of overwintering, we've done studies where we've dissected their guts to see whether they're feeding or not. It doesn't appear to be the case. So if we apply like a systemic to a plant to preload the plant so that it has the insecticide in the plant, when it bites into it, it's just not going to happen. We see them use their muscles in their wings to break down, to give them enough energy to get out there, feed, mate, and then they start eating as they kind of sail off into retirement. So how are we going to get it into the body? So that really reduces a lot of the insecticide classes that we have. And really, the two groups that we see that are effective against this insect, even though there are other contacts on this list, are the organophosphates as well as the pyrethroids. And not all organophosphates are going to be overly effective against these insects. So, you know, chlorpyrifos, the lesser effect acephate, pretty much all the pyrethroids work about the same. And we know that we have some issues with the pyrethroids as well. So those are really our two options. So if we're thinking about rotation, we're limited there. I will say we don't need to think about rotation within the season on adulticides. I think this is a one and done thing. Should be something that we focus only against the overwintering adults and then be done for the year. These are flamethrowers. What you get that day is what you're going to get. So you need to make sure that everything goes perfect for this application because it doesn't have residual activity. These things are going to wear off immediately almost. In our studies, what we've seen is residual activity drops dramatically after 24 hours, and we're getting very, very minimal control after that. Now, you might see some labels that have some neonicotinoids. For those of you who still can use neonics, not our friends in New Jersey or New York. But it's really, these are often in combination with one of these other active ingredients or other classes that is doing the killing. We don't see great impact on adults like we would with something like a bill bug. All right, so one of the studies that I mentioned about our egg laying studies, we also tracked how long these things live. So these are just survival curves that you can see. And what we did is we placed mating pairs into conical tubes to see really how long these things live, how long they're out there, but also how many eggs they produce over their lifetime. So males and females in the first year really kind of declined at a linear rate. Again, these are protected from birds and predators, so it might be an overestimation of actually how long they live out there in the world. But what you can see is they're capable of living a very long time, so between 11 and 14 weeks. Now, if we couple that with what the female is doing, and we did this by checking what they had laid eggs into the tillers each week and then moved them to a fresh set, we could see that the females were laying a variable amount of eggs every week. What we see in the black line here is temperature accumulation, so growing degree days in that week. So when the temperature is increasing, they start to increase their egg load. If it drops, they can't lay as many eggs. And what we can see, the main point of all of this is they're laying most of their eggs within an 8-week period. So again, if the goal is to reduce egg laying with this adulticide, I think it really needs to be well timed at that peak before they drop their eggs to reduce it. Otherwise, if we're solely using larvicides, whatever resulting from this egg laying, just know that you're probably going to have eggs enter the system for a long period of time. Now, it might not be 8 weeks. Again, this is ideal situation, but even if they're laying eggs for 3 weeks to 4 weeks at a time, that's a whole month where every week you have non-susceptible stages inside the plant emerging and damaging the crown in that week. All right. So let's look a little bit at the adulticides, pyrethroids. If you still have these available to you for efficacy, or you still have a susceptible population, this is by far the most efficacious of the two classes. If we look at the attributes, this is an exercise that I lead my students through in class. Can we look at some of the characteristics of these insecticides and then make hypotheses about what it would be able to control? So with the pyrethroids, we have low solubility, not a big problem. It's going to mix well in your tank, well enough anyway. But it has high binding, a high KOC value, meaning that once we apply it, it's not going to move down through the soil. It's going to bind very tightly to organic material. So it's never going to control something like your root feeding white grub with it. The low residual means it's going to work on contact as well. It's not going to continue to give. So our stem borers that are in the plant are not going to be exposed to it. They're going to be protected from the plant, which really leads us to use this product primarily against surface active insects. So of the pyrethroid uses, it's really going to be against those adults. Because it has such low residual activity, we need to optimize when we apply that, meaning when are the insects are at their peak density on the fairways or other short turf areas. Now, the big problem with the pyrethroids is they were too efficacious, I think, and probably too cheap. And when you have something that's really good and really cheap, you're going to use it a lot. Unfortunately, it has low residual, and that's going to cause people who say it's cheap and effective, I'm just going to apply it more frequently. And that's exactly what we saw in the 90s when this came onto the scene. What we could see, if you read I remember reading literature when I was in grad school in the early 2000s, and there was mention of people just using one pyrethroid application for the year, and that was good enough. It controlled their population so well, that was fine. There were other people that I interviewed in my time in graduate school who were making a pyrethroid application every 2 weeks for upwards of 10 weeks because they were so cheap and yet so effective. So it's really no surprise that we developed resistance in about a 15-year period after they were released in some populations. So overuse, we really want to limit the adulticide application to one application and be done for the year, in my opinion. Other people might use these summertime adult peaks to apply as well, but I think to make these things last as long as you can, keep it to one. And why I say that is it is pretty risky strategy, because what we're seeing in populations where they have developed resistance to pyrethroids, unrelated compounds become less effective as well. We think that this is a general mechanism to resistance. However, that might not be the case in all populations. But if it is a general mechanism of resistance that confers resistance to other insecticides, then all of a sudden, a lot of larvicides don't work, and you're really in a bind for what you can do to control this insect. All right, so when I talk about resistance, it's not a black or white thing. It's not a light switch on and off in this case. Resistance is a continuum, meaning if you've ever applied a pyrethroid, you're at some point on this arrow moving towards resistance. If you've never applied one, you might still have some susceptible stages out there and control is perfect. If you've inherited a property where you've applied a lot of pyrethroids throughout the year for various pests, maybe caterpillars, ants, and whatnot, you're constantly exposing these populations. You're basically increasing the probability that you have resistance in your population, so you're further on this line. So we can have different levels of susceptibility to these other compounds as well. All right, so the best practices with the adulticides, again, these are limited residual flamethrowers. What we want to do is wait until all those insects are out of overwintering and apply them when they're at maximum density. We do not see egg laying prior to this. We do not see any major feeding that would cause turf damage. They'll notch the plant. It's all superficial damage that's going to mow off. I'm not concerned about that. We have done studies to determine when the insect might be on top of the canopy, when it's most likely to come in contact with that insecticide. That's roughly between 57 and 64 degrees. However, I can tell you, I don't know anybody who holds off their spraying for adults until it's 57 to 64 degrees. So that might just be an academic exercise. And I'm not even sure that it would result in significant differences in that. What we can do is we can move the product in contact with those surface active insects. I would keep it light. I would keep my watering of these products light. I think I like to irrigate everything we put down. A light watering, you know, 1/10 of an inch maximum, a couple of spins of the head is fine for these adulticides. We don't want to move it too far, but it will bind to organic material. So in theory, the insect's going to be moving in and out of the thatch as it becomes active throughout the day. We have been doing a lot of work with surfactants and improving control. It seems like our quick wetters, alkyl polyglucosides combined with a block copolymer, there are some products that have those blends. Those seem to be very, very effective in improving movement, most likely through that waxy cuticle and into the insect. But the important thing about the adulticide, I think it's a very important application. I would like you to minimize that, keep it to one and done. All right, monitoring, I'm gonna cruise through this quickly because I don't want to maximize the time here. I want to get through to Lisa's stuff, far more interesting stuff. We have a couple of measures that we can use to time our adults. Soap flushing, I have it here on this slide as being the easiest. This involves dish soap and water buckets. It is fairly easy. I would say it's more economical. Vacuuming is another option that you can do, slightly harder, only because you need to have specialized equipment, but you can collect the adults much easier. Other things that are more passive using plant phenology, forsythia, half green, half gold plant development is correlated with the same time when insects or the ABW are at their maximum density on fairways. So if we time it like that, it takes a lot of guesswork out. Weevil track, which Lisa will speak about, is using researchers' boots on the ground to kind of take all of these measures together and then input what we're seeing in the region. Can also get you in the ballpark as well. And that's also using some growing degree days with it as well. Now moving on to larvicides, this is opportunity 2, you know, the 1-2 punch as I call it. You know, some people make 3 applications, an early larvicide and a soil larvae application. You know, this is a little bit more difficult to time than the adults, but we do have some good options for that, at least in springtime anyway. When we look at the menu of larvicides, it looks great on the surface. Instead of 2 classes, now I have, you know, 4 to 5 classes. But within those classes, we don't see a whole lot of active ingredients. So it's really kind of limited. It creates some rotational issues. I think people tend to stick to what they like. But the ones in yellow, I think, are the most efficacious classes, or the ones that we have the most evidence for at the moment. This should change in the later part of the class today, where we'll see another class come on. But essentially 5 actives prior to what we learned from Lisa. All right, so the challenges, again, we need to have a product that has translaminar activity or it's systemic. I don't see great activity with the systemics unless they're congregating in certain parts of the plant or have residual so that when the insect pops out of the plant, it's in place and the larvae comes in contact with it. The other consideration that we need to have is, did you apply an adulticide? Do we need a longer duration? If not, you know, we're going to be thinking about probably making more than 1 larvicide application. If we're solely relying on larvicides, we're probably going to need a couple to cover our bases over that long period of time. Sampling is a little bit harder for larvae. It involves things like taking a cup cutter, you know, putting in a salt solution, irritating those larvae out, waiting for an hour, making a mess in your office. But we can do things to better time when they are entering the soil, at least in the springtime. Once the spring generation is over, we have to do those more complex means of scouting. But this is hybrid rhododendron, produces a nice purple flower when this plant is in full bloom. This is anecdotally been reported to be the same time when the larvae are leaving the plant and entering the soil. So very reliable in our cases, in most places throughout the Northeast. So this is one that you could time these larvicide applications with. Now, My final kind of wrap a bow on all of this is the timing of these larvicides. Again, it's kind of tricky. It's more difficult than timing adults, where we're waiting for a peak. We're waiting for a population to move through when most of them are present in the soil. All right, so this was a study led by Albert Koppenhofer, and we had data in our courses in Pennsylvania on the susceptible population. We had populations in New Jersey with resistance population. I just want to show you the effects of resistance development, pyrethroid resistance development, on unrelated compounds such as larvicides. Now, typically we recommend this period here. So this would be when the larvae are exiting the plant. And you see that the insecticides that we had at the time, or the appropriate ones, all seem to do fairly well. The younger ones inside the plant, what we see here is Ference is providing very good control. I think it's the nature of the product, where it really accumulates in the crown where it's going to feed. It's probably getting these soil larvae later. Just this timing looks awesome. What we can also see is once we get too late, once the larvae get too big, and there's probably some pupae in the population at this point, all controls start to decrease to somewhat. Now that's 90% control. All right, so again, control is not perfect. Now what happens when we have a resistant population? All those bars get much higher. They look a lot like the untreated controls, and or at least statistically similar in some cases. But what we can see is once we develop resistance, now we're limited to only a couple of insecticides. Things like Ference do very well. Matchpoint is another one that does very well. But again, what we can see is there's the effect of timing. As these larvae get bigger and bigger, they're going to be harder to control. All right, so that's the end of my portion. Now we're in for the super fun stuff that Lisa will provide. And I will mute myself.
Okay, thanks, Ben, for that introduction. And I'm really excited to introduce to everyone some of the latest innovations coming down the line for control of annual bluegrass weevil coming from Syngenta. And so I'd like to take a minute here to, let's see, my controls are up. Okay, there we go. To talk a little bit about a new innovation coming from Syngenta, a new active ingredient that we're all very excited about. The active ingredient is plinazoline technology, and so the chemical name associated with that material is isocycloserum. And so upon federal and state registrations, which we expect to have very quickly, very soon, it will be marketed as a texo-insecticide. And so what's really exciting about this material is the chemical class is an isoxazoline, and this represents a new mode of action for us in the turfgrass industry, and so that's very exciting. And so it classifies into IRAC group 30, which is officially recognized as a GABA antagonist. And so I'll go into this a little bit more on the next slide. Essentially, it interferes with the nervous system in insects. And so our mode of entry, or the way that this works on pests, is via contact as well as ingestion. What we anticipate is no signal word associated with this material. It would be formulated as a 200 suspension concentrate. And so as a new mode of action, it's important to talk a little bit about how this material actually works. And so we all have a good understanding of that. And so you know, the nervous system is important in insects for sending signals, and one of the messengers that does that and is responsible for that signaling is called the GABA molecule. And so that sends nervous signals throughout the insects. It's a messenger signal. And what we find with plinazoline technology is it binds to a site that receives that messenger in the nervous system. And so what's important to recognize here is that there are other insecticides that do interfere with that GABA messenger, but the binding sites are different. And so that's how we have a new mode of action here with this chemistry. And so essentially what it does is it binds to an entirely different binding site than, say, something like fipronil or abamectin, and it interferes with the relaxation of the muscles. And so it stops them from relaxing, resulting in insect death over time. And so what we have is a product that affects adults and larvae. It is a broad spectrum on several insect pests as well as mites. And because this is a new mode of action here, we have no known cross resistance to other insecticides. And so I want to take just a moment here to talk a little bit about what insecticide development has looked like over the past several years here. And so, you know, and this is, if we think about in the turfgrass industry, we have really not seen a new mode of action in a number of years. And so the last development there would have been in the anthranilic diamides, or things like acelepryn and ferrants that came out in the early and mid 2010s there. And so this is something we're very excited about, to have a new mode of action coming our way here in a very short period of time. And so this is something that has been under development for a very long period of time. And just to give you a little bit of history here about plinazolin technology and the actual molecule and what a timeline of development of something like this looks like, is it's really been going on for 20 years. And so some of the early work in this IRAC Group 30 within Syngenta began around 2005, where there were some very early projects looking at several compounds in other projects. And from that process, we were able to select some promising molecules that were passed along into future development. And so you can kind of see there the timeline of how this takes shape. And so it's really a significant amount of time and energy and resources that goes into something like this. And so you can imagine why there is such excitement here in bringing this material to end users. So what are some of the properties here of this particular molecule, and what do we see with its behavior? And so some of the physical chemical properties are fairly unique. And so this is a material with very low water solubility. It is lipophilic, so it does bind to the soil organic matter. So essentially, it does not move once it is placed. And so you could see that here in these images here, looking at the photos on the right here, where you compare it to something like a neonic thiamethoxam that is moving through the plant after it's placed at the petiole, whereas plinazolin technology really stays where you put it. And you see the same in the leaf image below as well. And so, you know, we have a very strong binding potential to the leaf and the plant once it is put out. I mentioned it is a contact and ingestion material, a new mode of action in this IRAC Group 30. There is feeding cessation that results in insect mortality. We do have a residual that sticks around because this material binds so well and stays where you put it. We'll talk a little bit about the spectrum on the following slide, but I do really want to focus on ABW as the purpose of this webinar today. But you'll see a little bit about some of the other pesticide controls as well. And so we have very good crop safety. I think to date, we have hundreds of turf trials where we have not seen any phytotoxicity, very good UV stability as well on the leaf surface. And so this material really is something that sticks when we put it down. Okay, so a little bit of an overview of what a proposed label will look like upon a federal registration. And so I've mentioned several of these points already. IRAC Group 30, contact and ingestion, no signal word is anticipated. Our use sites would be golf courses, commercial turf, no residential turf at this time, sod farms and athletic fields. And so here you can see some of the other pests here that will be on the label, but for the purpose of today, I'll focus on annual bluegrass weevil and really highlighting some of the data and some of the work that we've done with this insect to date. So I'd like to take a little bit of time to talk about some of our trial data summaries and really highlight the efficacy of this molecule here. And one of the things that I have alluded to and mentioned is that we have control of adults and larvae. And so that is in fact the case when we think about a pest like annual bluegrass weevil. And so what you're looking at here are lab bioassays that were completed by Dr. McGraw and Dr. Kostromyska as well at the University of Massachusetts. And this is looking at overwintered annual bluegrass weevils. So these were collected in the field. They are exposed to the insecticide in a lab bioassay, and we look at mortality over time. And you can see here in both of these trials, we really have excellent adult mortality as we monitor these populations over time. And so this is pretty exciting to have a new adulticide coming down the pipeline. And so, you know, this is not something we test just in the lab. We also do this in the field as well. But as you can imagine, ABW are mobile, right? So this is a tricky thing to test in the field. And so the way that we typically do this is with caged studies. And so ABW are collected. Oftentimes they are placed here, as you see in this photo, in some sort of PVC type tube, and those tubes are covered so that we don't have birds or predators eating the insects, and they are treated in that way, and we come back and look at the number of surviving adults here. And so what you can see in the top graph here is comparing planazoline technology to Scimitar, and we have an excellent control of ABW adults 8 days after application. But what I think is really remarkable here is if you come back and look at the larvae in those plots as well, 40 days after that initial application, and we're not finding any larvae in the planazoline treated areas. So if we think about timing this in that early egg hatch to early larvae instar timing, as you heard about earlier, and this would really reflect for those of you that are weevil track followers, it would be the stage 2 of weevil track, our current stage 2, and this is what you're looking at, a summary here of 5 trial sites. The bars behind each of the dots are the average, and so you're looking at the number of larvae per square feet. And so really excellent efficacy from planazoline when applied at that timing. But of course, you know, we want to look at this and evaluate this at all timings. And so, you know, looking at this later timing, so when those larvae are a little bit larger on their way outside of the plant. And so this would coincide with what is the current stage 3 of weevil track. And at that timing, across these same locations, we also see excellent control with planazoline technology of those larger larvae. And so, you know, it's not always that we get to see nice photos of annual bluegrass weevil trials. And so I wanted to take a moment to highlight a demonstration trial that I was a part of this year in New York State. And this is a pure stand of annual bluegrass turf where planazoline was applied at the 8.2 fluid ounce rate on May 30th. And then nothing else was applied to this area. And so you can certainly see here our untreated area here in the foreground, and just the amount of feeding that is really coming up in that area. And sure, you know, there is a little bit here showing up in this area, but nothing else has been applied to this area for 62 days. And so this is really quite remarkable when you look at this type of image. So I think, this lends itself to the question a little bit of, with a new product coming to the market, how would this impact something like Weevil Track, and a program that has really become the backbone of annual bluegrass weevil control, and how would we see some changes possibly taking place. And so I want to just take a few minutes to talk about Weevil Track a little bit, and how it is important to those of you in the industry. And so this is a survey that was conducted in 2019. And so we had asked, what are the top 3 tools or indicators you rely on the most to monitor ABW? And you notice here that Weevil Track is really pulling a large chunk of the percentage of responses here. And so, this is a really valuable tool and a valuable resource to a lot of end users in the industry. And so some of the history behind Weevil Track, it was launched in 2009, and around that time there was 1,100 registered users or so. And right now I think we're sitting around, I pulled the data in June, around 3,600 registered users. And so there's certainly been an increase. In 2013, it was relaunched by Syngenta, and there was some user interface updates added, as well as scouting videos, and the ability to follow multiple courses. That's when we saw the addition of ferrets come in. And then from 2017 to present, really it was a lot of user experience updates. We expanded the number of cooperators that are part of Weevil Track, and that's when we saw the Weevil Track guarantees sort of enter the marketplace as well. And so it's a very useful tool, and with this tool, we can learn a lot, and we can look at this data and recognize some patterns in the data here. And so I wanted to include this because, I knew that Ben was speaking about the spread of this insect, and where it is moving in the United States. And I think one of the interesting things is, if we look at the Weevil Track subscribers, we can see changes in the subscribers over time here as well. And so here's our subscribers from 2020. The darker shaded areas are where we have higher concentrations of subscribers versus 2025. And so when I look at this, there's a couple of things that stand out here. And so you see more heavily shading in 2025 in the Midwest, which I think reflects that spread that Ben was speaking about earlier. And of course, California and Texas as well. And so if we break this down, and if we look at the states with the largest increase in Weevil Track followers, it really follows movement of this pest, as well as an increase in the insect monitoring sites that we even see in places like Kentucky. And so it's quite remarkable in my mind that we can use a web-based approach to sort of follow the insect pest and its movement in the United States. One of the other interesting things that I do not have a good idea as to what's going on here, a large increase in subscribers in California and Texas. So there's either some folks that are just very interested in annual bluegrass weevil, or perhaps, anticipating one day that they may see this pest. And so I think this speaks to the value of Weevil Track and the utility that end users see from this. And so, we have historically had an excellent group, and we still do, of cooperators that are really the boots on the ground for this, right? And you see all these folks here who contribute to blog posts and do the scouting, and really transition those stages for you. And so what you have seen for a very long time was the dedicated efforts of these individuals that have resulted in, these scout for stages, transitioning to stages that have directly related to a control strategy. And those stages have been closely tied to insect behavior, and then recommendations for control applications as well. And so, as you can imagine, and are probably thinking about, what may happen with Weevil Track as planazoline technology comes to the marketplace. And so what we will see in the next couple of slides is an anticipated or a proposed change that we may see in the coming months or so to Weevil Track with this new mode of action, and that includes planazoline technology that allows for enhanced efficacy, more flexibility, fewer applications, a simplification of this program overall. and a new approach to resistance management with a new mode of action. And so this would be planned as a relaunch to what we would be calling Weevil Track Plus. That would really be a significant update to this website that has been around since 2009. And so, you know, if we think about plinazolin technology, where would we see this fitting? And so this is a highlighted table here that is an attempt to break this down for you. And, you know, I've shown you with some of the data that we have excellent adult control data. And so really the nice fit for this product is here in this early timing that we would call phase 1, that targets overwintering adults and early instar larvae. And this would coincide with what we would see as Forsythia full green. And so this timing is a little bit different than what we have previously utilized in our Weevil track timings. Following that, to control those larger larvae as they come outside of the stem, Ference is an excellent fit in that timing as well for larval control in that overwintering first generation. As we proceed into the summer timing, and typically this timing, you know, we've heard about what a jumbled mess it becomes, so it could be anywhere from late June to early July. We have another fit for plinazolin technology, and as we progress later into the summer, again, Ference as well if needed. And so, you know, sometimes it's easier to break this down by exact phases, as I would call them. And so, you know, in this phase 1 stage with plinazolin technology, what would we be targeting? And we would be targeting overwintering adults, ABW adults, and young larvae. And so if we think about monitoring indicators, what this essentially translates to is it's about 7 to 10 days after that peak activity is observed, Forsythia full green, and on about, I'll give you a range here of growing degree days. It's about 160 to 250 growing degree days. And now I think as you've heard about some of these indicators, these passive indicators like growing degree days and plant morphology, these can vary across all locations, right? And so it's important to make sure that you're monitoring multiple plants, you know, you're monitoring growing degree days across multiple locations, and really nothing would replace actually scouting yourself. And so that's just a couple of watch outs there as we think about monitoring for this timing. And this is really, as you've heard, potentially the most critical application of the season. And so really what this allows us is to have a contact and an ingestion option against the adults at this early timing. So following that would be this phase 2, or targeting crown feeding large larvae. And if we think about the monitoring indicators there, this is when you can find larvae in cup cutter samples. It's a little more challenging to find them then, but this really relates to that hybrid rhododendron full bloom. Eastern redbuds are well as another good choice, and the growing degree days shift a little bit later here, and this could be anywhere from 350 to 450 growing degree days depending on your location. This is where damage can be extensive, especially if we see drought. And if there is adequate moisture at the time, you know, that can mask the damage there. But when we think about insecticide control options at this point, really the diamides or products with systemic activity such as Ference make a really good choice here. And so what would something like this look like visually if we were to compare this overwintering ABW control strategy from the Weevil track that we're all very familiar with to what we would call Weevil track plus? And really what you're seeing here is this reduction and this is our original Weevil track optimum control strategy that many of you are likely very familiar with to this point. And you see these 3 applications of Scimitar, Acelyprin, and Ference. At those 3 timings that were referred to as stages. And what would be different here is that this timing of plinazolin technology would shift a little bit further down the time scale to target those overwintering adults as well as early instars, and then followed by a Ference application down the line. And so, you know, this is something that has been thoroughly tested, and we have looked at this quite frequently throughout different locations and trial sites. And you can see here again, the 5 different trial sites we've tested at, and the means and the bars in the back. And you can see that this program that I just described, featuring plinazolin and ferrins here, providing excellent control. If we were to average that across all of those locations, it's 95% control, compared to our current weevil track optimum control strategy at 87%. And so the program works very well. It reduces the number of applications and it gives enhanced control. And so what happens if you miss that timing slightly, or something happens and you aren't able to make those applications? Well, we've done some work looking at rescue applications for annual bluegrass weevil control with plinazolin as well. And so this particular study, conducted by Steve McDonald, went out when the larvae were much larger, 4th and 5th instar larvae. And you can see at the time of application, you know, 60% of the larvae in there were 4th instar larvae, and this area received 1 application of plinazolin and did very well 12 days after application at reducing those larvae down to almost nothing in those plots. And so that's that really money shot that we want to target there, is that first overwintering generation. But, you know, as Ben mentioned, there could be options later down the summer where you have breakthrough or you have needs for control as well. And so this is where we think of this summer generation or second generation of ABW control. And this would translate to a phase 3 or phase 4 of Weevil Track Plus. And so really monitoring indicators, continued scouting is key. I mean, as Ben alluded to, this is a jumbled mess. Generally, you start to see second generation adults emerge sometime in late June, early July, but these populations can be spotty. They might not be all in the same area. They might not be as dense. But if we do have a very hot summer, damage can be severe. And so at these stages, then you have a new option. You have plinazolin technology for both contact and ingestion, and ferrins as well for that systemic activity at this timing. And so visually, again, to give you an idea of what this looks like, the current Weevil Track program is up here on the top of this chart. And so, you know, we had stage 4, stage 5, and stage 6 and stage 7 that were in there. And really, we will see this condensed with the anticipated launch of Weevil Track Plus down to plinazolin technology that would fall in this late June to early July timing, followed by ferrins at this late July to early August timing. So really a significant reduction here in the number of applications. And I want to take a minute here to wrap up to talk about insecticide resistance, since this was something that Ben had highlighted in his talk. And so this is something that an area that we are actively pursuing and interested in, and especially with bringing a new chemistry to market, this is something we always want to look at and evaluate. And so we have done field testing of against pyrethroid resistant annual bluegrass weevil populations. And so this is a study at a site that has 55x resistance to bifenthrin. And so I would say that that is a good example of a golf course with moderate resistance to pyrethroids. And so with one application here applied in late May, really excellent control of those populations. But, you know, when we think about any type of resistance, whether it's insecticides or fungicides, you know, one of the things that's important to us is that we don't just look at this in the field, we also look at this in the laboratory. And so we are also invested in resistance monitoring, and this is something that has been going on behind the scenes as we prepare to bring plinazolin to market. And really what we do is we look at various insect populations. In the case of ABW, what we have done is selected insects from 6 unique geographic locations, and those insects are exposed in the lab to a serial dilution or very, very small amounts of the actual active ingredient that is applied via a very tiny syringe to the back of their prothorax. And so it is dropped on them at different serial dilution concentrations ranging from nothing to high concentrations. And we look at their mortality over time. And what this really does is this allows us to look at the number of ABW that are pyrethroid resistance that responds to this chemistry, and allows us to establish a baseline so that we can monitor resistance in the future, as well as determine how well our technologies work against these populations. And so this is something that we've been invested in and is continuing behind the scenes. And so we should have some more to share on that in the near future. And so with that, I think I just want to mention that, coming down the line with the federal and state registration of plinazolin technology, there will be a new Weevil Track Plus guarantee coming. And for those of you that are still and very familiar with our current Weevil Track guarantee, don't worry, that will still be fully supported as well. And so that is still a viable option out there as well. I just wanted to, make that clear to everyone on the call. And so you can look for this coming soon. And then, I just kind of want to mention again the tools available to you on Greencast Online. So I'm sure everyone is familiar with Weevil Track and where to get that information on Greencast. However, there are many resources available there to you. I've highlighted a few that have been very valuable to me over the years. And so make sure you check that out and stay in touch with the website for the latest updates on this particular chemistry. And so with that, I would be happy to take any questions, and I'm sure Ben would as well, or if Ben would like to chime in on anything. I know I showed quite a bit of his research. He's welcome to chime in as well.
Thanks. Lots of details, lots of great info there for people to use. We really appreciate that. There is a question. Ben talked about 2 key timing points to control ABW. Since Lisa mentioned that peazolon controls adults and larvae, and through contact and ingestion, is it safe to assume that helps with greater flexibility with your application timing while maintaining efficacy?
Who wants to handle that? I think Lisa probably sees more of the data. She does see more of the data than I do, so I'll let her handle that.
Yeah, I would say yes, that there is a greater flexibility in there with what we see in terms of the timing with plinazolin. I still very much like that early timing just to target that adult activity that we see in that generation coming out of overwintering. But we do know there is flexibility there and that you could time this at other timings if needed.
Yeah, and I'll add my own observations that we haven't done field experiments with adults. We did do the adult lab assay, so it does look broadly toxic. It looked really, really good against early instar larvae, so the ones in the plant. So suggesting, translaminar activity. I think we can safely say that based on kind of the tech sheet on that. So yeah, I think you do have some adults out there at that time too. So if you were not making it an adulticide and you were timing it with those early larvae, you'd probably still be knocking back some of the adults as well.
Okay. If you have other questions, now's the time to send them in. I do want to mention that Ben is going to be teaching in February. It's right around the corner, folks. Conference and show. We're already selling out some of the seminars. The last report we got, we had 2 sellouts. So come and see Ben teach. Our next live webinar, John Fetch is going to talk about telling your story, why bother? And one of the things he's going to do is use some video clips. So if you have joined using the app, you might want to think about just using your browser because they, you know, internet rules, they don't let you just use things. So the way the recordings work with the app is not real compatible. Doug has his hand up. I'm going to unmute. And Doug, if you really have a question, I've unmuted you. You can ask that now. Sometimes people click the raise hand when they didn't mean to, but. Well, Doug, we're not hearing anything there. If you figure out that unmute, let us know, or you can type that in.
Can you hear me
There
now?
we go. We can. What's
We
up?
can.
Super. Hey, so quick question. If you have treated turf, do you know how many feet the adult weevil would need to transverse through treated turf to pick up enough AI to be effective?
Oh, I don't think anybody on planet Earth knows that. Maybe they do, Doug. I love talking with Doug. He always has great questions. I would say maybe you're getting to the point of like banding, like maybe if I'm jumping too far ahead.
No, you got it.
Okay, so like the idea of banding, you know, people have tried this anecdotally. I don't think there's ever been any studies that have looked at it in depth. I've seen some recent grant proposals looking at it. But the idea would be to treat like a swath, maybe a boom width or two on the edge of fairways.
Okay, thank you.
And I think like in theory, it should work. We have seen it break down in the field where we've had non-replicated kind of observational stuff where it seems like they've gotten into the middle of it. So in theory, I love it. Largely untested though.
Yeah, so we have sometimes tried that with the adult studies we've done, right? Where you introduce them to an area and you band around that in hopes that they stay in your trial site. It's not always successful. So they do still find a way to avoid that and get out of those areas.
I would also add that, you know, adulticides are often short-lived. And some insects, you know, there's evidence in like turfgrass ant where they can sense residues and kind of can delay it, their activity based on that. So that could be what is at play there. I mean, really, if they delayed any sort of activity for 24 hours, the rate of control would go down dramatically.
Okay. Other questions, raise your hand or type them in. The deadline for the nutrient survey has just been extended to November 20th. So if you have not filled that out yet, please do so. That helps us to be able to advocate. Also, there's Centennial merchandise, Ben. They've got some really cool new stuff. It's available online. We will be bringing it to conference, but some of the things are maybe going to sell out. There's one or two items that are starting to, as I'm told, have a little bit lesser inventory. So if there's something you see that you really want, you know, it'd be sad if your Sterling Santa Fe mug wasn't there for you.
I was looking at the Santa Fe mug.
I liked that one. There are way more things. These are just the ones that I thought were, you know, interesting little variety. We do have an exit survey as you leave today. So if you would provide that feedback, your education points will be processed. No further action is needed for that. Any closing remarks for us? Because I see no further questions. Nope, nothing further. All right, we'll stand adjourned. Thanks so much, Ben, for teaching. Lisa, we appreciate you. All the team at Syngenta. And I know I speak for everybody when they say we're looking forward to that new product and seeing how the Weevil Track Plus is working. Less resistance. Always a good goal, right? All right, everybody.
Thanks,
We'll
Lisa.
stand adjourned. Thank
Thank
you. Have
you.
a great day.
You too.